All right, now, as we dive into Mendelian genetics, we're going to be learning all about the different types of crosses that we do in genetics. For example, if mom has a certain hair color and dad has a certain hair color, what are the chances that the offspring will have a certain hair color? You guys all did a really good job with your Mendelian genetics projects where you went back and kind of revisited the history of Gregor Mendel. We want to start by remembering that Gregor Mendel was an Austrian monk. But let's go back to his original pea plants to start with, and let's talk about the flowers. So, for example, he had some flowers and he had some pea plants that he was experimenting with, and he had a group of flowers that were purple. Let's say he had 10 flowers, and he went ahead and bred those or crossed those with another set of purple flowers, for example. And he was a Austrian monk working in the monastery, not necessarily a scientist by trade, although that really did evolve later on for him. So his hypothesis was that we would get all purple flowers. It makes sense, right? Purple flowers crossed with purple flowers. Maybe we'll get purple flowers, but this was where the laws of inheritance came to play. The idea of a dominant trait versus a recessive trait. All right, so that was our starting point. Again, you guys did a good job with those presentations. And let's dive into the cell, okay? Take a moment real quick, answer this question. Where in the cell do we find the DNA? All right, so yes, the DNA would be found in the nucleus of the cell, and even more specifically than that, it's going to be found on what is called a chromosome, all right? And those chromosomes are basically coiled up DNA. In your written notebooks, if you have not done so already, please make a new section for Mendelian genetics. You should already have some introductory heredity notes in there. All right, and then the name of today's day would be inheritance patterns. Do the best you can here to try and draw this. I want you to be able to see that we have, first of all, the structure of a chromosome. Don't worry about the labels, but to start with, let's focus on drawing a chromosome in our notebooks. All right, so the chromosome itself is going to have pieces of DNA wrapped up in it. That kind of think of it as a spool of yarn, and we have the individual pieces of yarn that come off would be the DNA. And as you can see, here's our full DNA structure right there. So I just want you to get this basic idea of what the chromosome looks like and see how the chromosome is that DNA supercoiled and wrapped up. So go ahead and get that drawing done. And we are now going to take a look over here at a little more detail of the chromosome. So, for example, if this is the chromosomal material from the male, and this is the chromosomal material from the female. Of course, in plants, they have both. So these are what we call homologous chromosome pairs. All right, so on each individual piece of chromosome, there's going to be what we call a locus, okay? And a locus is going to simply be the location of an allele on a chromosome. And we're going to dive into what an allele is here in a moment, but first let's understand that the locus is the specific location on that chromosome where a specific allele is. So, for example, this is my complete chromosome. I may have right here a locus, okay, and that locus might be where the genetic information is for, say, hair color, okay, so that's where the allele is going to be located for hair color. All right. Let's get into some of the vocabulary, guys. You're going to want to make sure you get good definitions down of these things. There is a lot of vocabulary in genetics. Once you get it down, it makes everything just that much more easier. So an allele is a variation of a trait. So if the trait is fur color, then there's going to be various alleles, obviously, for fur color. The locus, as we just said, is the location of an allele on a chromosome. And then a chromosome is going to be basically the coiled, or we'll say supercoiled DNA that holds all the genetic info. Right, so looking at the different types of inheritance patterns, what does that mean? So these are going to be the different types of crosses, basically, that we're going to be doing. So the first one, so this would be complete dominance, and this is basically what Gregor Mendel came up with in his garden, right? So we have one trait that is going to completely mask the other trait, hence the name complete dominance. So as you're solving genetics problems, the first thing you're going to need to do when you read the problem is try and identify what type of an inheritance pattern is it. Is it complete dominance, or does it fall down here to an exception to complete dominance? So for complete dominance, we have basically only one trait, okay, or more specifically, one allele will show in the offspring. Okay, so this was what we saw with the pea plants. All right, so with the pea plants, the pea was either going to be smooth or wrinkled, right? The flower was either going to be purple or white. There was no in between, okay? The types of crosses here that we're going to be learning are going to be monohybrid and dihybrid, where monohybrid are simply going to be the crosses between one trait. So mom has brown hair, dad has blonde hair. What will the offspring look like? Or what are the percentages for the offspring? Dihybrid would be two traits. So what's the chance that we have blonde hair and green eyes at the same time, for example? So this is going to be complete dominance. We're going to go through some example problems here shortly so I can show you how to solve each one of these types of problems. Exceptions, right? So in biology, there's always some exceptions to things. So we're going to take a look at exceptions to Mendel. So for the first one, we're going to have codominant. So this is going to be an example where we're going to see both traits show through. Okay, so there's going to be two alleles that are seen in close to an equal amount on the offspring. All right, so we're going to be, for example, here we could have a black chicken crossed with a white chicken makes a speckled chicken. So if this was complete dominance, it'd be either the black trait or the white trait would be dominant, and that would show through in the offspring. In codominance, both are going to show through. So a speckled chicken is going to be, for example, a chicken that has both black and white. I have a picture of it here for you to show you. So this is my best codominant example here, all right, where we have the black chicken, the white chicken makes a speckled chicken, okay? So what does this mean? What is this going to look like, okay? We're going to dive into that in more detail here in just a moment once we go through our complete dominance. All right, so that is my example here for codominance. And then incomplete dominance is going to be like red flowers. It's going to be a mix of the two. Red flowers and white make pink. All right, so a mix. And again, these are exceptions to what Gregor Mendel originally found out. And then sex-linked traits simply are going to be things like color blindness, all right, where it shows up on the X chromosome. And we'll dive into those as well. Before we go into doing our first complete dominance cross, and again, this lesson is going to stop after complete dominance, and then we have another lesson coming up on the exceptions to Mendel. So once again, by the end of this lesson, you should understand complete dominance. You should understand how to do a monohybrid cross, a dihybrid cross, have an understanding of a pedigree, and then some of our basic vocabulary like allele and locus. So we've got a pedigree, which is basically a genetic family tree. We're using squares for males and circles for females. As you can see, if the square is completely shaded in, then it's going to be an affected male, which means it has or shows the trait or the disorder that we're talking about. A lot of times when we use a pedigree, we're talking about some type of a disorder that we might be mapping in a family tree. Like, is somebody a carrier for color blindness, or is somebody a carrier for sickle cell anemia, for example? We usually don't use it for something like, what's your hair color going to be, that kind of thing, okay? If it's a circle, it's again, and it's completely shaded in, it's going to be an affected female. If it's half shaded in on either one, it's going to mean that it's a carrier. And then if we put a slash through one like this, it would mean that that person is deceased in the family tree or the pedigree. All right, so here's an example right here. So whatever this trait is, it could be sickle cell anemia or whatever. You can see here that the dad has it, the mom's a carrier, and that gives a really high chance that the offspring will be carriers. This is why a lot of people do a DNA test before having kids, to kind of see some of the possibility of these things. All right, so this is, again, just what a pedigree is. We're going to spend a lot more time on pedigrees in the coming weeks, especially as we get into evolution, as we look at how traits are passed on from generation to generation. All right, now we are going to start the process of solving these problems. So first of all, I would like you to get this basic skeleton down on the steps for how to solve genetics problems. For example, here, this monohybrid cross. So step one, I want you to show the genotypes of the parents. And then in red here, I have our first practice problem. We're going to be crossing a heterozygous blue-haired frog with a homozygous recessive pink-haired frog. Sounds about right. And then the second step is going to be to complete the cross. And then we're going to write the answer, which is going to always be the genotypic and phenotypic ratio. So go ahead and get these steps down, and then leave 4 or 5 spaces under number 3 down here, so that I can go ahead and we can get the rest of your vocabulary down. So to make it easier for us to solve some of these problems, let's just double check that we've got our vocab down. I think you guys probably already have a pretty good understanding of these terms. The first one, looking at our genotype and phenotype, where the genotype is going to be the combination of the alleles. So if big B is the dominant trait there, it's going to mask that recessive trait, that little b. The phenotype is the physical characteristic. So big B, might be brown hair, blonde hair, okay? In this case, we'll say it's brown hair. And we have our dominant and recessive alleles. So make sure that you're clear on those 4 terms. Homozygous and heterozygous are going to be important as well, as homozygous is going to be when we have 2 of the same alleles. So if it's homozygous dominant, then that would be big B, big B. If it's homozygous recessive, it'd be little b, little b. And you can see for heterozygous, we're going to have big B and little b. So taking a look now at solving our first practice problem with our heterozygous blue-haired froggy and our homozygous recessive pink-haired froggy, the first step is to show the genotypes of the parents. So my heterozygous blue-haired frog, all right, we're identifying that blue is dominant, is going to be big B, little b, again because it is heterozygous. Our homozygous recessive pink-haired frog, all right, is going to be little b, since it is homozygous. Okay, this is why that vocabulary is important, because you're going to be utilizing it in these problems. The second part here to complete this cross, to actually complete it, is we have one parent here. So my first frog alleles are going to go on the top. My second frog alleles are going to go on the side, like this. And I just simply go through and do my cross. I'm sure you've probably done something like this before. Alright, and then the last step is to go through and do the genotypic and phenotypic ratios, which sometimes they're the same, sometimes they're different, but we're still going to want you to write these out. So we have a big B, little b. This is going to be 50%. Little b, 50%. Then my phenotypic ratios are going to be the same. So for blue hair, it's going to be 50%. And for pink hair, it's also going to be 50%. Okay, so that's where we're at there, solving our simple monohybrid cross. Okay, take a quick inventory of our learning thus far. So this was lesson number 1. Well, actually, lesson number 1 was the heredity intro. All right, so you have that, your notes from that. So this technically would be lesson number 2, and we call this inheritance patterns. And our focus was the vocab, the structure of a chromosome, and monohybrid crosses, all right, for complete dominance. Okay, so this lesson really is focused on complete dominance. The next lesson in the next class that I'll be recording here shortly will be focused on the exceptions to Mendel. So that's going to be incomplete dominance, codominance, all right, and sex-linked traits, and then also our dihybrid complete dominance. At this time in your OneNote, you're going to want to go to the pre-IB biology, the 2021 one, not the second semester notebook, but go to the regular notebook, and you should be able to complete this monohybrid cross worksheet. Just go through it as a practice. You don't need to turn this in, but this will be a good little practice review for you to finish now that you've completed this activity here. So these things also will be on Canvas. This first one right here, these genetics practice problems, these will be due. You will have to complete these and turn them in, but those will not be due quite yet.